In this talk, we will learn about different strategies that are used to find when organisms evolved, followed by the phylogenetic species concept. The discipline of systematics studies the diversity and evolutionary relationships, or phylogeny, among different organisms. These evolutionary relationships are pictorially depicted using phylogenetic trees. In case you were wondering about the etymology of phylogeny, it arises from the word philo, which means tribe, and geny, which means generation. The phylogenetic trees can include not only organisms that are presently living or extant, but they can also include extinct organisms. Cladistics is a method of classifying organisms where the primary criteria is based on their common ancestry. Using cladistics, phylogenetic trees are built based on identifying characters that are similar between different organisms. These characters can be the presence, absence, or loss of certain traits or features. For example, one character can be that the organism has a body that shows bilateral symmetry, which means that there is a line that can divide the body into a left and a right part that are mirror images of one another. Another trait could be the absence of hair. For example, fish don't have hair. They have scales instead. Similarly, the presence of hair can also be a character. Mammals tend to have hair on their body, and hence it is a good character to define mammals. The loss of a tail can also be considered as a character because, as we have seen in humans, we know that our ancestors had tails, but we don't, and hence apes lost their tail. Thus, we can have different types of characters that can be used to build these phylogenetic trees using the discipline of cladistics. The characters that are used to build phylogenetic trees can be derived or ancestral. Shared derived characters, which are also called as synapomorphies, are characters that are present in all members of a group and they evolved from the most recent ancestor. For example, if we consider the group Mammalia, we know that its common ancestor evolved the character of producing milk. Thus, milk production is a shared derived character for the group or clade of Mammalia. Similarly, if we consider the group that includes Reptilia and Mammalia, their most recent ancestor evolved the character of producing eggs that have an amniotic membrane. And hence, the production of an amniotic egg is a shared derived character for the group that includes Reptilia and Mammalia, and this clade is called Amniota. Similarly, if we now consider a group that includes Amphibia, Reptilia, and Mammalia, their most recent ancestor evolved limbs that had digits. All the organisms in this group now have limbs that have digits, and in fact they have 4 limbs. And hence they all come under the group of Tetrapoda, and the character of having 4 limbs with digits is the shared derived character. Shared ancestral characters, on the other hand, are called simplesiomorphies. These are characters that arose prior to the most recent ancestor of the group. Going back to our group of Mammalia, we agree that the production of milk is a shared derived character for the group. However, the production of an amniotic egg, which evolved in a more distant ancestor, would be considered as a shared ancestral character for the group of Mammalia. If we consider the group of amniotes that include Reptilia and Mammalia, in this case, The evolution of the amniotic egg, which was observed in the most recent ancestor, is the derived character. However, evolving limbs with digits, and especially having 4 of those, would be an example of a shared ancestral character. This is because this character was evolved in a more distant ancestor. Similarly, if we look at the group of tetrapods that includes amphibia, reptilia, and mammalia, then the character of limbs with digits is a derived character. However, the character of having lobed fins would become an ancestral character. Thus, we can see that characters can be ancestral or derived depending on the context. Thus, by using different characters and determining whether they are derived or ancestral, we can create groups that include a common ancestor and all the descendants of that ancestor, whether they're living or extinct. Such groups are called as clades. Phylogenetic trees that show the relationships among different clades are called as a cladogram. Now, it's important to note that a clade can include one species or a group of species. As long as the clade includes the common ancestor and all of its descendants, it satisfies all the conditions to be defined as a clade. Such clades are monophyletic in nature. It should be noted that if any of the descendants are not included in the group, then that group cannot be called as a clade. In such situations, that group is a paraphyletic group because it includes the common ancestor and only some of the descendants. Another situation can arise where a group can be defined which includes certain descendants but does not include the ancestors. Again, such a group is not a clade, and in fact, this is a polyphyletic group. Thus, to be considered as a clade, the group has to include the common ancestor and all the descendants of that ancestor. A phylogenetic tree or cladogram shows the evolutionary relationships among different organisms. They are comprised of branches and nodes. The nodes denote recent and distant ancestors. For example, when we look at the group of chimps and humans, the node that is shown represents the direct ancestor or the most recent ancestor. On the other hand, other nodes that we can observe show more distant ancestors for the group of chimps and humans. Branches, on the other hand, show species at the end of them. The location of a species or a branch can be revised based on the information that is available because each branch is considered as a hypothesis. Thus, it is a prediction of the evolutionary relationship of that specific species in relation to the other species present on the phylogenetic tree. The length of the branch can indicate the genetic difference or evolutionary changes that happen between the organisms and their ancestors. Generally speaking, the longer the branch, the more the evolutionary change between the organism and its ancestor. Phylogenetic trees can be constructed to not only show the different evolutionary relationships among organisms, but they can also depict when the organisms evolved. In such situations, the timeline when the ancestors or the organisms themselves evolved is provided on one of the axes. In this case, it is provided in the x-axis. To find out when organisms evolved, different approaches can be used. For example, fossils can be analyzed. Based on the availability of tissues in the fossils, radiocarbon dating can be performed to know the age of the fossil. If the fossil is too old and thus out of the range of radiocarbon dating, then the rocks that were associated with the fossils can be analyzed and thereby we can estimate the age of the organism. A more recent approach that has been used in order to understand when organisms evolved is the use of a molecular clock. A molecular clock is based on the rate of change that occurs in either DNA or protein sequences with time. The molecular clock hypothesis is an idea that states that some DNA sequences can undergo mutations at a constant rate over time, and this can occur among different organisms. Not all DNA sequences can function as a molecular clock. Only the genes that change due to genetic drift, or mitochondrial DNA, or pseudogenes, can function as a molecular clock. The mutations that usually happen in these DNA sequences normally do not affect the fitness of the organism, and hence these mutations or changes that happen in the DNA sequences are considered to be neutral mutations. These mutations usually come in the form of base substitutions. The different steps involved in building a molecular clock include identifying an appropriate DNA sequence and then sequencing different organisms to determine how many mutations occurred in that specific DNA sequence. The next step would be to determine the age of these organisms, and this can be done usually through dating the fossil. A curve is then generated by looking at the mutations that happen in the different organisms along with the time at which these organisms evolved. This allows the generation of a mutation rate curve, which tells us how many mutations occurred with time. Using this curve, we are able to now estimate the age of other species by knowing how many mutations happen in their DNA sequence. For example, if we have an organism that had around 25 nucleotide substitutions in the DNA sequence that we are considering, for example, cytochrome C, as shown in this figure, then we can now estimate the age of that organism to be around 30 million years. And that way, the organism evolved around 30 million years ago. Such a curve has to be generated for each DNA sequence that will be considered as a molecular clock. Thus, molecular clocks provide timelines to when new species were formed. The genetic difference between two species can be considered to be proportional to the time since they shared a common ancestor. It should be noted that in order to use a molecular clock onto a species whose age is not known, it has to first be validated by applying it to the organisms for which we know the time of evolution. It should also be noted that molecular clocks can be used not only with DNA sequences, but also with protein sequences. While building phylogenetic trees or cladograms, similar characters present between different organisms are taken into account. There are some aspects that can cause building these phylogenetic trees more challenging, and one of these is homoplasy. Homoplasy is when there are similar features that are independent of descent. Homoplasies can occur due to sheer coincidence. For example, we can have a situation where the nucleotide sequence between two organisms are quite different, but there are some areas where we do see similarities. This is an example of a molecular homoplasy because these similarities or identical regions occur due to simple coincidence. On the other hand, a homoplasy could be a product of convergent evolution. For example, if we take the wing of a bird and the wing of a butterfly, they are analogous structures. They are performing the same function, which is that of flying. However, their anatomical features are completely different, thereby indicating that do not have a common ancestor, and hence arose through convergent evolution. It should be noted that analogous structures should never be taken into account while building phylogenetic trees. Horizontal gene transfers can also cause challenges while building phylogenetic trees. When we have the inheritance of genetic material from parent to progeny, that is an example of vertical gene transfer. In the case of horizontal gene transfers, genetic material is obtained through other sources rather than inheritance. In this example, we are able to see a horizontal gene transfer when a virus has infected a human and has incorporated its genome into that human. Horizontal gene transfers can cause changes in phenotypes. For example, pea aphids are usually green in color. However, some of them obtained genes from fungi that code for carotenoid molecules, and such types of aphids now have a pink color. If we use the color of the aphid as a character, then we would consider the green and the pink colored aphids as two different species, which is not the case. And thus, horizontal gene transfer can sometimes cause challenges while building phylogenetic trees. One of the most widely accepted definitions of a species is the biological species concept, which states that a group of organisms that have the ability to breed with one another constitute a species. If they're unable to breed with other animals, then those animals are a different species. The problem with this explanation is that there is an emphasis on sexual reproduction. There are many organisms that do not utilize sexual reproduction, and hence classifying them into their appropriate species becomes difficult with the biological species concept. Additionally, it does not take into account that we could have organisms that are able to breed with one another but cannot because they are present in different habitats and hence have been considered as different species for now. With the advent of DNA sequencing and the ability to build more accurate phylogenetic trees, the phylogenetic species concept has been proposed, where a species is defined as a group that has one or more shared derived characters. The phylogenetic species concept addressed some of the shortcomings of the biological species concept. It places no emphasis on reproductive mechanisms, and hence it can be applied to organisms that show sexual or asexual or both modes of reproduction. It also does not take into account location of organisms. One of the main criticisms, though, is that it may result in different species that have only slight changes in their derived characters. If we look at these aphids, for example, and consider body color as a shared derived character, then they would be considered as different species, even though they're not, because the pink-colored aphids are showing that color because they obtain genes from a fungus through horizontal gene transfer. Hence, phylogenetic species concept is not perfect, but another way to define species. With this, we come to the end of our talk where we learned about phylogenetic trees or cladograms and how they depict the evolutionary relationships among different organisms. We learned about accounting for time in phylogenetic trees and different approaches used to accomplish this. We learned about different factors to consider while creating these cladograms or phylogenetic trees, including horizontal gene transfer and homoplasies. We finally learned about the phylogenetic species concept and how it was able to address some of the shortcomings of the biological species concept.